Here, we will describe how to generate a cell aggregate solution using either ReLeSR or gentle cell dissociation reagent, GCDR. PSCs are ready to be passaged when the majority of colonies are large, compact, and have dense centers compared to their edges. Also, it is common to see colonies merging together at the time of passage. Start by examining the morphology of the cultures under the microscope. Using a phase contrast microscope, begin at low magnification and scan the well to assess the quality of the PSC morphology. To reiterate, at the time of passage, PSC colonies should be relatively large, cells should be tightly packed, exhibit phase brightness at the center of the colony due to multilayering, and have a distinct border. It is also normal to see colonies merging at this stage. Moving to higher magnifications, healthy PSC colonies will possess prominent nucleoli, have a high nucleus to cytoplasm ratio, in that the nucleus will practically occupy the entire cell, and the tight cellular packing should be even more evident than what is observed at low magnification. If you passage using ReLeSR, begin by washing each well with 1 milliliter of PBS without calcium or magnesium. Aspirate the wash solution. Now add 1 milliliter of ReLeSR to the well, and aspirate all of the ReLeSR within 1 minute. It is not recommended to leave any ReLeSR on the cells for the upcoming incubation time. Then, incubate the plate at 37 degrees Celsius for 5 to 7 minutes. If transitioning from a medium other than mTeSR1, this step may have to be altered. Incubation times will need to be optimized for each cell line to ensure that areas of differentiation are not brought forward to the next passage. If you find areas of differentiation are being brought forward to the next passage, reduce the incubation time by 1 to 2 minutes. Once the incubation time is over, remove the plate from the incubator and add 1 milliliter of mTeSR1 or mTeSR Plus to the well. You will immediately see the undifferentiated PSC clumps lift off the bottom of the well. To detach the undifferentiated PSCs, place the plate on a plate vortexer for 2 to 3 minutes at 1200 RPM at room temperature, or manually tap the side of the plate using the base of your palm for 30 to 60 seconds. Using a 5 milliliter serological pipette, transfer the detached cell aggregates to a 15 milliliter conical tube. Examine an aliquot of the aggregate suspension under a microscope fitted with an ocular micrometer to determine the size of the aggregates generated. Mean aggregate size should be between 50 to 200 micrometers in diameter. If the majority of the aggregates are above 200 micrometer, triturate the aggregate suspension by pipetting the suspension up and down with a 5 milliliter serological pipette until the desired aggregate size is generated. When passaging using GCDR, you will need to assess the morphology as described above. At low magnification, if there are suspected areas of differentiation, in that there is a loss of border integrity, cellular packing appears more loose and disorganized, if there are visible holes or areas of non-uniformity within a colony, investigate by moving to a higher magnification. At a higher magnification, it should become obvious if the colony contains differentiated cells. These cells will typically appear larger and less tightly packed than healthy cells in a colony. Once you have identified an area of differentiation, using a microscope object marker or a permanent marker, mark the area on the underside of the plate. When the entire well has been assessed and areas of differentiation have been marked, move to the biosafety cabinet to begin passaging. Using a sterile P1000 pipette tip, scrape the areas of differentiation as marked previously, and aspirate the spent medium and the differentiated cells that you have scraped with the pipette tip. Add 1 milliliter of GCDR to the well and incubate at room temperature for 6 to 12 minutes. The incubation time may vary between cell lines and matrices. Dissociation should be monitored under the microscope until the optimal time is determined. The optimal incubation time is reached when increased light and gaps appear between cells located on the edge of the colony. If the culture is under-incubated, the colonies will not be dissociated sufficiently. If the culture is over-incubated, large holes will be evident, and upon scraping, many single cells will be generated rather than aggregates. Aspirate the GCDR and add 1 milliliter of mTeSR1 or mTeSR Plus to the well. Using a cell scraper, scrape the cells from the plate, ensuring not to splash the medium from the well. Now, transfer the detached cell aggregates from the well to a 15 milliliter conical tube using a 5 milliliter serological pipette. Then, carefully pipette the aggregate mixture up and down to break up the aggregates as needed. A uniform suspension of aggregates approximately 50 to 200 micrometers in size is optimal. Examine an aliquot of the aggregate suspension under a microscope fitted with an ocular micrometer to determine the size of the aggregates generated. Mean aggregate should be between 50 to 200 micrometers in diameter. If the majority of the aggregates are above 200 micrometers, triturate the aggregate suspension by pipetting the suspension up and down until the desired aggregate size is generated. Now that you have created a cell aggregate mixture using either ReLeSR or GCDR, it is time to plate them for the transition to mTeSR+. When transitioning from mTeSR1 or other feeder-free media to mTeSR+, it is important to maintain parallel cultures in the original medium until mTeSR+ cultures are established. Label a Corning Matrigel or Vitronectin XF-coated 6-well plate, as illustrated here. Add 2 milliliters of the appropriate medium to each well of the plate. So for wells in column 1, add 2 milliliters of mTeSR1 or whichever feeder-free medium is currently being used. For wells in columns 2 and 3, add 2 milliliters of mTeSR+. Plate the cell aggregate mixture as follows. For wells in column 1 and 2, plate the aggregates at a density or split ratio currently being used. As some cell lines expand better in mTeSR+, you may notice better attachment, such that a lower seeding density is necessary to prevent overgrowth. Therefore, for wells in column 3, plate the aggregates at a seeding density that is at least 25% less than the current seeding density. Then place the plate in a 37 degrees Celsius incubator with a 5% CO2 and 95% humidity atmosphere, and move the plate in several quick, short, back and forth, and side-to-side motions to distribute the cell aggregates. Do not disturb the plate for 24 hours. For the next step in the process, proceed to video 3, Maintenance and Morphological Assessment.